Now we're going to look at the last pathway, or the third pathway in those three-link pathways that are responsible for metabolizing monosaccharides, then extracting electrons and protons from them, from the intermediates in the pathways, and they are transported then to the electron transport chain. And it's those reduced coenzymes, NADH and FADH2, that are responsible for carrying those electrons to this respiratory chain in the mitochondria. And these are reducing agents, so they can be oxidized, and that's what happens when they enter that, or when they reach the electron transport chain. So NADH is oxidized back to NAD plus and FADH2 back to FAD, so they can go back and pick up more protons and electrons. The electrons that are transferred to the electron transport chain are passed or migrate from complex to complex and undergo a series of redox reactions, which produces some energy. Ultimately, at the end of the respiratory chain, the electrons and hydrogen ions react with O2 molecules to form water. So thus this process is aerobic because of the use of that molecular oxygen in the transport chain. So we're going to look at a diagram or a depiction of the electron transport chain that's embedded in the inner membrane of the mitochondria. And one of the functions of this pathway is to pump hydrogen ions across that inner membrane. And the buildup of those hydrogen ions in that inner membrane space is going to create what we call a gradient. So we're going to have a higher concentration of hydrogen ions in the inner membrane space than we do in the matrix, which is within that inner membrane. And what that does is it creates this potential energy gradient. You may have heard of something called the chemiosmotic theory, and essentially this is the model of what happens in the electron transport chain, where that hydrogen ion gradient is built up, and that eventually drives the synthesis of ATP. So those hydrogen ions in the inner membrane space have traveled from the matrix either by going through a channel or being dropped off or transferred to one of the four complexes in the electron transport chain. But ultimately, when we get to a certain concentration in that gradient, the ions start traveling back through a channel, and this channel is called the ATP synthase enzyme. And this enzyme is responsible for producing ATP. And of course it does so by phosphorylation of ADP to ATP. So we've got a constant pool of both of these molecules in our system, and a way to convert ADP to ATP when we need more. And we'll talk about how much we need on the last slide. So the energy released as those protons return to the matrix is the energy stored in that ATP molecule. And this is called oxidative phosphorylation, because the energy used to transfer the phosphate group results from the oxidation of the coenzymes, NADH and FADH2. So you saw this slide, and we're almost done. This is a short video, the very last one for this semester. When we introduced metabolism, and I just want to go over this again before we look at the detail, or some of the detail. We aren't going to see as much as you could see with respect to that electron transport chain. But remember, we looked at the structure and the organelles that are within a cell. And we focused on the mitochondria because those are considered the powerhouses in the cell because they produce ATP. It has the outer membrane, the inner membrane space, so this is where that proton gradient builds up. This is the inner membrane, which is very invaginated, lots of folds, and there's the matrix within that space inside that inner membrane. So the electron transport chain is actually embedded in that inner membrane. And you see that 3 of these complexes, 1, 3, and 4, this is the ATP synthase, actually traverse the entire membrane. So they have a little piece sticking out on this end of the enzyme or protein. And these are the 3 complexes. Complex 2 doesn't contribute to the proton gradient, but 1, 3, and 4 pump hydrogen ions across that inner membrane space, or inner membrane, into the inner membrane space. This is the matrix side of the mitochondria. So those NADHs and FADH2s that were generated by the combination of glycolysis and the citric acid cycle migrate to the inner membrane, inner side of that inner membrane in the matrix, and they get oxidized, and their protons or hydrogen ions get transferred to inner membrane space, and the electrons are transferred to other redox centers that we don't see that are within these complexes. So ultimately, these electrons are transferred to other electron carriers. Their electrons transferred here. Notice there's no hydrogen ion, but these hydrogen ions do come off, and actually, let's say that these are those hydrogen ions right there. But electrons do get transferred and transported or migrate or are carried to the 3rd complex, the 4th as well. Continuous transfer of hydrogen ions across that inner membrane. And here's the oxidative phosphorylation, where we have molecular oxygen being converted to water using some of the protons that are present. And when this gradient reaches a certain threshold, then protons enter the ATP synthase, and this is a very complex enzyme, but it is responsible for phosphorylating that ATP or ADP to form ATP. So now I want you to think about a few things, and I have some questions here on the next slide to kind of recap. Oh, I'm sorry, one more slide and then the questions, or maybe 2 more. We've seen this information before. NADH entering the electron transport chain produces enough energy to make 2.5 ATPs, and FADH 1.5 ATPs. So that's what? 4 ATPs there. The citric acid cycle produces 3 NADHs, which is 2 ATPs, or equivalent to 2.5 ATPs. So we get 3, or 1 makes 2.5. Sorry, so we get 7.5 overall. FADH, 1.5. We can have a GTP produced in the citric acid cycle. So that's 10 ATPs that they contribute to, in summary, citric acid cycle. So a very serious consequence of some drugs and chemicals, basically chemicals, if any one step of the electron transport chain or oxidative phosphorylation is disrupted, the organism cannot survive because it can't make ATP. Hydrogen cyanide is an example here. It produces the cyanide ion, which irreversibly binds to the iron 3+ portion of cytochrome oxidase. So this is one of the enzymes in the complex, or one of the complex, and it goes back and forth between Fe3+, and Fe2+. So it is one of those acceptors of electrons that are carried to the electron transport chain by those reduced coenzymes. So it goes back and forth between 3+ and 2+. In this direction, it's accepted an electron, and when it transfers it to another redox center, it will go back to a 3+. But if this gets irreversibly bound, it's not able to pick up an electron. So that electron transport down that chain doesn't happen. So cytochrome oxidase is a key enzyme of the complex 4 in the electron transport chain. There are other substances that might partially impede the electron transport chain. So it really depends upon what the substance is, whether it will be a complete loss of ATP production, in which case the organism will die or the cell will die, or just a partial disruption of ATP synthesis. So here's the last slide. Just some questions I wanted you to think about. I am going to give you the answers, but I wanted you to think about them in terms of what is going on, and we've only looked at one pathway that takes place in the cells of our bodies. Number one, how many cells are in your body? It's estimated that in the average human body, there are 37.2 trillion cells. That's a pretty big number. Very big. How many mitochondria are in each cell? And it really depends upon what kind of cell we're talking about, because some tissues might require more energy than others. So for example, muscle and heart would need more ATP than, an epidermal cell, for example. Also the brain, and we're going to take a look at that too. But on average, there are about 2,000 mitochondria in one cell. Okay. I couldn't find a number for how many electron transport chains would be found per mitochondria. I don't know why I couldn't find that, but I didn't find anything that I felt was reliable enough to write down. Sorry, I can't write with my mouse very well. But we know there's likely more than one embedded in that inner membrane. How much glucose does the body metabolize daily? And on average, and this too depends upon your size, but on average, about 200 grams of glucose are metabolized daily. That means that, no, that's not what I was going to say. Never mind. 200 grams of glucose daily. What I was thinking of was the molar mass of glucose. That's about 180 grams per mole. So that means we're burning or metabolizing, or the average human is metabolizing a little over a mole of glucose. The other thing I was going to tell you here was the brain metabolizes 60% of that. So 60% of the glucose that the body metabolizes in one day is metabolized by the brain. It needs a lot of fuel to run our body. How much ATP does the body use daily? Okay, so we metabolize these monosaccharides in order to produce ATP. And also remember that we've just focused on carbohydrates, but amino acids get metabolized, triacylglycerols get metabolized, so they too are going to contribute to the production of acetyl-CoA, and thus can enter the citric acid cycle and also contribute to the production of ATP. So depending upon the tissue, you might be metabolizing triacylglycerols for ATP. But last question, how much ATP does the body use daily? And that is estimated to be about 100 to 150 moles. If I can write that. I'm working on my desktop today and that I don't have a touch screen here. 100 to 150 moles of ATP per day. That translates to 50 to 75 kilograms of ATP. So essentially, your body uses a mass of ATP in one day that's basically equivalent to your body mass. And that means that those cells in your body are working hard every day, producing ATP, and also conducting all the other processes that happen in them, which we haven't even begun to touch on in this course. So maybe this will spark an interest in you to look at some of the other processes that occur here or in your cells that They're basically minute chemistry factories, constantly working and running reactions. So this is the last video, as I said, but I will be meeting with you next week.